Hello everyone, and welcome to week 1 of the Clinical Laboratory and Application Diagnostic Procedures 2 lecture. This will be a continuation of what you had in 1460, or if you haven't taken Procedures 1, this will essentially be us getting to know different sections of the medical world. So for Module 1, we're focusing on what we call the clinical laboratory, and that will be Chapter 34, page 733, Chapter 44, pages 1039 through 1043, and all of Chapter 47. Now these are all units that you have discussed, especially if you took fundamentals already. Definitely things that you've learned on, so it'll be more of a review than really a new learning. So let's go ahead and get started and talk about each of these. We're going to start with Chapter 34, which is talking about infection control. And the one area that we really focus on for this is going to be that chain of infection. And the chain of infection is how pathogens and organisms get transferred from one host to another. The chain of infection does have 5 links. Those are your reservoir host, portal of exit, means of transmission, portal of entry, and susceptible host. So your reservoir host is really the beginning. This is where a host, either an animal or a human, gets infected with a microorganism. Then we move to the portal of exit, which is when the microorganism leaves the reservoir host and finds a new host to infect. It can leave through sneezing, coughing, sexual orientations, and sexual transmission. It could be an open cut or an open wound. Any of those can be a portal of exit. Your means of transmission is really how the organism travels. It's either going to be a direct or an indirect travel. Your portal of entry has to be able to get into a new host, so it has to, however it left the body is also how it has to get into a new body. And then your last one is your susceptible host, which is when a new host has been infected with the organism. Now, as a medical assistant, it really is your job to stop and break the chain of infection. And this can be done through proper hand hygiene, wearing your proper protective equipment, or PPE. So that is Chapter 34 for us. Let's move on to Chapter 44, which is talking about the clinical laboratory itself. And what we're focused on with the clinical laboratory is really the idea of quality assurance and quality control. Now, quality assurance is a way for us to really understand the how a process works. So quality assurance is carrying out procedures which involve planned and systematic activities to ensure that requirements are met and results are accurate. Quality assurance involves routinely reviewing procedural manuals, following the most recent manufacturer instructions for correct test performance. Now, part of that will also include maintenance, and all laboratory equipment must be maintained on a regular basis according to the manufacturer's instructions. And you also want to document all the importance for quality assurance. So that means making sure that you're documenting your control logs and making sure that you have reported maintenance checks. Quality control is a piece of quality assurance that focuses on the processes to ensure quality. It's really focused on the physical proof that results are accurate. Some machines require evaluation by calibration devices, which are specialized prepared strips or cartridges that are designed to produce a predetermined result. Or they can use control solutions, which are chemicals that produce an expected result that are usually purchased from a provided manufacturer for testing the equipment. Quality control should be done every day on all devices or kits to make sure that they're appropriately working. Along with learning about the quality assurance and quality control, we also have to look at OSHA regulations as well as CLIA regulations. Now, OSHA was established by Congress in 1970 with the Department of Labor to create safeguards covering nearly every employee in the United States. Under OSHA, they came up with what we call infection control. So that's kind of what we talked about with Chapter 34. It's following all of those regulations, knowing the bloodborne pathogens, your universal precautions, your standard precautions, things that you had talked about previously in your fundamentals course. In 1996, the CDC developed and published specific guidelines for isolation precautions in hospitals, and these were termed what we call our standard precautions, along with the bloodborne pathogens, where you treat all human blood and bodily fluids as though they are infected. One that might be new to you here is the CLIA, and CLIA stands for the Clinical Laboratory Improvement Act, or amendment, that was created in 1988 by Congress. It was developed by the Center for Medicaid and Medicare Services in response to a widespread concern over the accuracy of test results. CLIA divides laboratories into what we call 3 categories. The test can either be waived, moderate complexity, or high complexity. Each category specifies the types of tests that may be performed, and medical assistants are only qualified to perform waived testing. Next thing that we need to talk about with this chapter is laboratory hazards. So laboratories contain biohazard, chemical hazards, and physical hazards. However, most accidents are preventable. Lab safety must be the concern of all who are employed in the laboratory field. Medical assistants have to be familiar with hazard communication standards, standard precautions in bloodborne pathogens, hazard waste operations, and needle stick safety and prevention. When we talk about chemical hazards, every chemical should have an MSDS, a material safety data sheet, to provide information on that chemical. The MSDS will provide identification, safety information about how to properly store and dispose of the chemical, any potential health hazards, and handling precautions, as well as fire and explosive information. With your bloodborne pathogen and standard precautions, this was brought in as of 1992. OSHA created them, and it pretty much states that you have to consider all bodily fluids as though they could be potentially hazardous and label them properly. Your needle stick hazards was a revision made by OSHA to the bloodborne pathogen standards. It was put into act in 2000, and it pretty much states that healthcare employers must follow certain protocols, and they have to review all new safety devices to lessen the needle stick risks of their employees. Annually, this will have to be reviewed, and they have to put into use, and they have to use the opinions of their staff as they're the ones who use them. Fire and safety hazards, you have to make sure that you know what your safety plans are, what your exits are, how to handle a situation if there is a hazard like this, knowing where your eye wash stations are, your emergency showers, your safety blankets, things of that nature. And then your hazardous waste removal, you need to make sure that you are putting all blood and blood products, bodily fluids, tissues, cultures, vaccines, sharps, gloves, inoculation loops, paper contaminated with bodily fluids, all of those go into the proper waste disposals. Okay? Whether that be a sharps container, a biohazardous container, whatever it might be, you need to make sure that it's leak-proof and puncture-proof, and it has your biohazardous label. All right, so that will end chapter 44. The last thing we're going to talk about is 47, which talks about phlebotomy and blood collection. So throughout this, you may hear me say phlebotomy or venipuncture. Know that it means the same thing. So your role during this is you want to properly collect, label, and process and store these specimens. You also want to make sure that in order to do any sort of phlebotomy, you have an understanding of the circulatory system, the equipment, the term asepsis, and the actual procedure itself. So with your circulatory system, it's important that you understand kind of the basics of how the heart, blood vessels, and blood itself works. The heart has one main job, and that is to pump blood out and keep blood moving. And then we have your arteries, your capillaries, your veins, your arterioles, all of those things. And essentially all of your blood vessels are a one-way track, okay? Your arteries will deal with blood and carry blood that goes away from the heart. And then your veins will actually be the ones that carry it back. They'll pick up some oxygen in your lungs, and then they'll return the blood back to the heart. Now your capillaries are more towards the top layers of your skin, and they act more as the gas station for your blood. This is really where they drop off or they pick up that oxygen and they dispose of the carbon dioxide. And they kind of act as kind of that mini layer or mini stop station in between arteries and veins. Your capillary puncture, I'm sure as you remember, are skin punctures. This is where we talk about getting small amounts of blood either from your fingertips, your heels, or your earlobes. Your venipuncture procedure, I'm sure many of you remember how to do this. Best place to actually do this is what we call the antecubital space or the bend in the elbow. And you have three methods of which you use: vacuum tube, butterfly, and syringe. We also deal with order of draw, and I'm sure that many of you remember the importance of an order of draw, knowing that you have to do this in order to avoid actually contaminating your tubes and ruining a blood sample. And some of the last things that we have to think about are some special circumstances or special challenges when drawing blood. Things such as a hematoma or a bruise can happen. Remembering that patients always have the right to refuse. If they do say that they do not want the procedure, do not go through with it. It can be considered battery if you do. Stress has a lot of problems and can actually affect a lot of results, especially if they have a pre-existing condition such as diabetes or seizures that may actually make your test worse. Maybe you fail to obtain blood, and that completely happens. Specimen problems, maybe you draw the wrong tube or you draw out of the wrong tube. And then the last one we have are blood culture issues. Alright guys, so for this week you have one homework assignment. It is a case study in which you are going to research methods of quality control and then in a properly formatted APA document, describe how a medical assistant would promote and practice these methods to ensure a positive outcome when performing a phlebotomy procedure. This should be about 1 page in length. Again, making sure you're writing in APA format. That means the cover page, references, in-text citations, and a reference page at the end. Okay? In your case study, you should also include a definition of quality control, the steps you would take to plan ahead for a phlebotomy procedure, how to properly identify a patient, description on how you would select appropriate location for a venipuncture or a capillary puncture, how to obtain a quality blood sample, and at least one special consideration related to the patient. If you have any questions or concerns, guys, please make sure that you reach out to me as soon as possible. You can either reach out to me via email in Blackboard, or my direct line is on your home course page. Have a great week 1, guys, and remember, I'm here to help you. Bye-bye.